All right, how many people out there are graduating seniors? Remember, when you get your degree sheet signed, put it in the mailbox because I have to sign all of them. And then when you hand them in to, if you're majoring in biology, that is, or biopsych or biochem. And then when you hand it in to the student services, make sure you hand in that new advisement report that has to go with it. So remember, all those spaces have to be filled out. If you don't fill them out, I do, so I don't want to have to do that. All right. All right, so we're going to continue to talk about negative and positive selection in the thymus, and then we're going to talk about activation of T cells, and not the signaling, we already done that, but what other situations are needed to lead to the activation and the signaling inside the T cell. Okay. And if you read the book I think is really good. I know people don't think it is, but it is. There's a lot of information in there, and again, I'm trying to help you see the forest from the trees in that textbook. There's a lot of nice, valuable information in there. And again, I'm only going to test you on the material that we cover in class, but I think, obviously, I know I can't cover everything in the text, and if you read the text, you'll get even more in-depth understanding of basic immunology than I can possibly do during a lecture. All right, so what we're going to do is this classic experiment. Again, I left off with this on Tuesday, and I can't believe it's Thursday already. And we're going to show, indeed, the thymus is where positive selection is taking place. So we're going to infect our mouse with this virus we've talked about a number of times, with the lymphochoriotic meningovirus. And this is our control experiment, and this particular mouse is an AB hybrid. So it has both haplotypes A and B, crudely shown here, just being capital A, capital B. You know by now we show them as lowercase, but let's just indulge the cartoonist here. And it turns out if we do that, when we take spleen cells and we have targeted or infected target cells, the spleen cells from this AB hybrid can kill either infected cells, which are just strain A, or strain B. Okay, so that's something which makes sense. But then let's do the experimentation as outlined in this cartoon. And we're going to take, again, the same AB hybrid shown correctly here as far as the haplotype A / haplotype B. Lowercase. We're going to thymectomize the animal. It means we actually took out the thymus, which is not an easy task to do. We're then going to lethally irradiate the mouse. And what that means is when you lethally irradiate a mouse, unless you rescue it, it will die of massive infection. So that's what that means. So lethally irradiating it destroys the hematopoietic cells in the bone marrow, and it essentially destroys also adult cells, one hopes, of the immune system, and therefore the animal is completely at the mercy of the microbial world. Let the animal recover in a sterile environment. So again, this is not an easy experiment to do. Then you graft a thymus from another mouse, which is only represents the haplotype little b. Just imagine what's going on here as far as procedure-wise. And you know, we can put this thymus anywhere. We don't have to put it back where it originally came from. We then transfuse into this lethally irradiated mouse rescue cells, hematopoietic stem cells from an AB hybrid. And then we infect the animal again, our experimental animal, as opposed to the control in the previous slide, with LCMV. After all that, we let the animal recover for a few weeks, and then we sacrifice it. After all of that, we take out its spleen cells, and then in culture, we expose those spleen cells, which we know by now in this course contains lots of lymphocytes. including CD8 and CD4 cells. And then we expose those cells, and again, we didn't enrich them for CD4 or CD8, but that didn't matter, to infected cells of haplotype A or of haplotype B. And you can see the results here, and I want you to discuss amongst yourselves what all these results mean and what this experimentation allowed us to understand about positive and negative selection. I want you to think about all the steps that were done by these authors who did win the Nobel Prize. Zinkernagel did, the leader of the lab, and figure it out. Go ahead. This is a nice experiment.
Yeah, so if you grafted cells with like a top or one
All right, what's the moral to the story of this experiment? What's the moral to the story of this experiment? Since it's an Aesop's fable, what is the moral to the story here? That, let's start with that word, that, Ilana?
That the thymus is what selects for which strain of cells are able to live and proliferate in the immune system, because even though it was an AB mouse and they injected AB cells, only because the thymus was strain B, only the B cells were killed.
That's right. So the thymus decides who shall live. The thymus decides, putting it more poetically, who shall live and who shall die. And as it is written in the thymus. And therefore, the B haplotype thymus will only undergo positive selection for these hematopoietic stem cells, which have T cell receptors, which are able to bind to the B haplotype. Okay. Those which can bind to the A haplotype fail by what? Die by death by neglect, another poetic way of stating it, and therefore never make it out of the thymus. And as a result, even though this mouse is a hybrid, its thymus is not. So now we know the thymus indeed is where this process occurs, absolutely, and the positive selection depends upon the cells that the thymocytes will encounter in the thymus gland. We now know positive selection can be affected by the cortical thymic epithelial cells, the CTECs, and negative selection to a large extent by the MTECs, as well as dendritic cells in the medulla. Okay, so this is a great experiment. Well heralded and alluded to and referred to, even though it's been done many years ago, all the time. Now this is review. We already talked about AIRE, autoimmune regulator, and this is a molecule which is upregulated in the medulla of the thymus, not in the cortex, but the medulla, and principally, if not exclusively, by MTECs, medullary thymic epithelial cells. Again, that's the jargon. And AIR itself isn't really a transcription factor, but it binds to closed chromatin and opens it up and allows RNA polymerase II to bind to many regions on the genome and therefore lead to transcription of genes you wouldn't expect in the thymus. I gave you an example, one used by many instructors, not just me, that insulin would appear in the medulla of the thymus. You wouldn't normally expect that to be the case. So certainly not produced by the thymus. And therefore, these molecules, if the thymocytes are exposed to them, they're eliminated because if they bind too tightly by a process of negative selection. So AIRE is really important. And remember yesterday I uploaded sample test questions with answers, and one of them I do explain AIRE. So it's really an important molecule to know. All right, so now I want you to figure this one out on your own. This is a fun cartoon. Go ahead and figure this one out. And yes, you will have things like this on the exam. I'll give you a cartoon. You have to tell me what it all means. Or I do reverse engineering. I tell you what it means, then you have to show me the experiment. Get it? All right. So we have forward engineering and reverse engineering. Go ahead. I'll shut up and go ahead. By the way, when you put your phones up here, because I'm OCD, could you make them parallel to each other? It just looks more attractive to my eye. All different positions drives me insane. All right, so go ahead. And as usual, I should have not had this little bit of writing here. Sort of gives it away. All right, first of all, what cells produce AIRE? Instantaneously, what cells produce AIRE? MTEX. Okay. Now, when you produce the molecules that the AIRE demands, the MTEX can present that molecule both as class 1 and class 2 because it's produced endogenously in the MTEC. Moreover, it's secreted and then taken back into the MTEC. These cells, even though they're epithelial cells, do undergo pinocytosis and endocytosis. They have both endogenous and exogenous presentation of antigens, class 1 and class 2. The dendritic cells, which are also in the medulla, can effect negative selection. They can take up all these molecules produced by AIRE and locally synthesized by endocytosis for sure. They'll present it with class 2. But when I say that we are eliminating cells in the medulla, we are talking about, in this case, MHC. And dendritic cells certainly express rich numbers of class 1 and class 2, and therefore they're perfectly capable, even without the MTEX, of bringing about negative selection against self-MHC molecules. So that being said, what the heck went on here? Experimentally, what were the steps done and what did they prove? Experimentally, you know these techniques pretty darn well at this point. Okay, someone raise your hand. Anybody? Going once, going twice. Sold for $5. Who did I see your hand raised? No one. Someone raise your hand. Anybody, raise your hand. Come on, raise your hand, somebody. Volunteer to tell me what happened in this slide. Yes, rescued at last. Go ahead.
After the bone marrow transplant,
By the way, what's this little doop, 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 doop mean? This little doop, 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 doop arrow mean? Lethal irradiation. This is the universal sign for lethal irradiation, okay? Zap to MHC haplotype A. Why does this animal die? Because we rescued it, put it in a place where there are no microbes in the environment, sterile environment. Okay, go ahead.
so yeah, after the bone marrow transplant, it looks like it could tolerate the skin graft from the mouse with the other haplotype.
Yeah, you wouldn't expect it, correct? Because if you took a skin from haplotype B and you put it in a haplotype A mouse, it certainly would reject it in a very short period of time. It doesn't. What does that prove? Ipso facto, that proves what?
That bone marrow cells also, well, it says negative selection, so.
That's right. And what are those bone marrow derived cells which affect or bring about negative selection? MTEC can also do it, but cells which can do it as well are dendritic cells. So dendritic cells migrate to the medulla of the thymus and bring about negative selection. In this case, we're talking about MHC molecules, which are richly expressed by dendritic cells. So that would mean if you don't have an identical twin or sibling, that would mean if you got lethally irradiated at birth and then you got a bone marrow transplant from your sibling, you would then be able to take a transplant later from that sibling because presumably you would be then tolerant of that foreign MHC. I wouldn't suggest that, by the way, for a variety of reasons, but it's something to think about. Don't do it. Don't do it. All right. All right. Think about this one. You're going to see to figure out this one on your own. HY antigens are only produced by the Y chromosome. It therefore goes without saying that if you do not possess a Y chromosome, you do not express HY proteins. And you figure out the rest. You certainly know what a transgenic animal is at this point, and about allelic exclusion. That's all involved with this experiment. Remember, T cells are always given two names, specificity and restriction. Restriction and specificity. I expect you know this notation is cold. And I want you to interpret everything, even though these are lousy depicting data because we only have pluses and minuses, but I want you to figure everything out. Why there's some more pluses and fewer pluses and that stuff. I want you to figure out everything. There's an explanation for everything on this table. volunteer to take us through all the outcomes, data-wise? Any volunteers? Surely. People who are still talking, I assume you're trying to figure it out. Any volunteers? All right. Which mouse is going to express the HY protein? The male mouse or the female mouse? That's right, because what does the male mouse have that the female mouse does not have? Y chromosome. What's the only chromosome which codes for HY antigen or HY protein? Y chromosome. All right, so we know that to be the case. Now, thymocytes, let's take it bit by bit here. We have 3 different mice here. We have a male B haplotype, we have a female B haplotype, and we have a female of a D haplotype. And by the way, capital D, where is this located? Centromeric or telomeric? You should know that too. Centromeric or telomeric? Telomeric, right? Is this class 1 or class 2? Ooh, I heard some 2s out there. You are so wrong. This is what? If you don't know that by now, that's really not good. What are the class 2? IA and IE. K and D are class 1. You must know that for next Thursday. If you don't, you're going to be in Dutch. That means in trouble. All right. Okay. Double negatives. Are they more or less the same? Don't worry about the double positive here. Are they more or less the same in all 3 mice? Yes. How about double positives? More or less the same in all 3 mice? More or less? Yeah, any problem with that? Any problem? How many phases, stages of the double negative do we go through? 4. Do we have to know what happens in each of those 4 phases? I told you no, but the double negative goes through many phases itself. There's many different molecules that are upregulated and downregulated. But at that point, the thymocytes are double negative. Has any selection gone on at the double negative stage? No, therefore you wouldn't expect any difference between these 3 strains of mice. All right, double positive. Double positive, what happens at the double positive stage? Do we start having our positive and negative selection? Well, positive selection in any case. All right. Also, negative selection can occur in the cortex, so both phenomena can take place. Remember, negative selection occurs in the cortex as well as the medulla, but more negative selection occurs in the medulla because of AIRE being expressed by the M-TET cells. All right? All right, someone explain this one. Explain this, explain that, explain this. Remember, what's the transgenic TCR? What's the restriction of the transgenic TCR? Class 1 or class 2? Class 1, you know that immediately because of H2D, okay? This is a sign for the yen. You know why that's up here? That's showing you there's no Y antigen. All right, anyway, I just saw that. All right, so why? Well, wait a minute. Didn't I tell you with the transgenic animal, by allelic exclusion, you shouldn't have anything except the transgene which was given to the mouse? Why do we have any CD4 positive cells? Shouldn't. You shouldn't. Yes, open that window because it's like 1,000,000 degrees in here. All right. All right, don't fall out, please. I don't want to see that happen. All right, someone tell me that. Why are there CD4 cells here? We're transgenic by allelic exclusion. All we should have is class 1 restricted, and we know class 1 restricted T cells are what? CD4 or CD8? CD8, right. So why are there CD4 cells here? Single positive. Okay, anyone tell me why? Remember, you're the experimenter. You get data. You have to figure out what's going on. Yeah.
Could the peptide be endocytosed?
Sorry, I can't hear you.
Could it be like digested by or endocytosed by cells?
No, nothing that esoteric. Is allelic exclusion absolute? No, nothing in nature is absolute except death. All right, and taxes, as I say. For some people, taxes, but everybody death. All right. You know, some people in this country don't pay taxes. At any rate, not me, I pay a lot. All right, my fair share. Allelic exclusion is not perfect. Allelic exclusion is not perfect, and therefore some kind of splicing escapes through, and therefore we get a CD4 cell, which is TCR, which is then class 2 restricted rather than class 1 restricted, because allelic exclusion is good, but not absolute. All right. However, someone explain this third one, this female mouse, which is the D haplotype instead of the B haplotype. She's a D haplotype instead of the B haplotype. Why is she a negative for CD4? It's fun with data, because she failed what? Not Bio 104, which I won't go into, but she failed what? She had death by neglect, because her TCR could not hook up to anything in her thymus because she was a D thymus, and the TCR was what restriction? B restriction. So all of her cells, essentially in this case, assuming she didn't have any kind of escape from allelic exclusion, died from neglect. All right? So that's a case of lack of positive selection for her. And what about the final one, CD8 positive only? Why is it negative, positive, positive, negative? Why is it negative, positive, positive, negative? Why is it negative, positive, positive, negative? All right, TCRs in this male were what? What was the specificity for the TCR in this male? HY. Does he express HY? Yes. Therefore, his thymocytes will be killed by what process? Positive or negative selection? Lack of positive selection are because of negative selection. Which ones are going to be killed by? Death by neglect or death by two are what? What's he going to die from? Too much binding, because he's going to express the HY antigen. The TCR has now the restriction and specificity that it recognizes in the thymus. The HY antigen protein will be expressed in the medulla, especially because of AIRE. And either and or MTEX and or dendritic cells in the medulla will then bring about negative selection of these thymocytes. So therefore, none will escape to the periphery. So he will be CD8 minus. CD8 positive. There will be none CD8 positive cells. The female will be unperturbed because there will be no negative selection against the HY antigen. Why? Because even though she has AIRE being expressed, does she have a Y chromosome? No. Therefore, she doesn't produce the HY protein, and therefore it's not presented to her thymocytes, and therefore no negative selection takes place. So she escapes. Our female, again, of the D haplotype, she fails everything because of death by neglect. Yep.
But in negative selection, don't we want binding that's not too strong?
No, you want positive. In positive selection, you want some binding, but not too strong. You want to bind with positive selection, but not too strong, because if you're too strong, then you die of negative selection. If you don't bind at all, then you die from death by neglect. So you want to bind with some kind of affinity, but not a high affinity.
Oh, so the fact that the male produces the H1 antigen.
Right, he'll bind too tightly, too high affinity, and he'll die. The thymocytes will die.
Okay, but like for example, for humans, don't we? So you pulled up like an example of insulin. So don't we produce insulin? So
right. So in that case, you're going to have negative selection. If you produce insulin via AIRE in the medulla, that'll be expressed by the MTEX and by the dendritic cells. and any thymocyte which is specific for any kind of part of the insulin protein will be killed off. They'll bind too tightly. Okay. Remember, you're looking for selection against self molecules as well as self MHC molecules. So it's a twofold process, or twofold destination or outcome. All right. Now here's three things. One, here's the second one. Two, by the way, what's the meaning of stochastic? Don't look it up. What's the meaning of stochastic? Anyone know the meaning of stochastic? All right, I'll give you a synonym. Yes?
By random chance?
Yes. Random is the way to go. Stochastic is by random. Okay, kinetic. You know what that means? What's the name that goes along with kinetic energy? What's the other type of energy? That's nothing to do with this at all. All right. Okay. Now, what we're looking at here is the instructive model, the stochastic model, or the kinetic model for how a double positive thymocyte ends up either single positive 4 or single positive 8. There are three different possibilities which are bandied about of how this takes place. One is called the instructive model, one is called the stochastic model, and one is called the kinetic signaling model. I know you've looked at all three of these before, but I'm going to help you with the first one, but not the next two. I want you to figure out the next two. The instructive model means randomly a thymocyte has a TCR, which is either CD4 or class 2, rather, able to recognize class 2 molecules, or just by accident able to recognize class 1 molecules, just by an accident. Okay? And when that happens, if the molecule it produces is able to bind to a class 1 molecule, then it downregulates CD4 and upregulates CD8. On the other hand, if it just produced a TCR, which can recognize class 2, it downregulates CD8 and only has CD4, and therefore it becomes a single positive CD4, and the one on top is single positive CD8. So it's just by accident what the T cell receptor produces when it splices VJ and VD and J for the alpha and beta chains respectively. And again, just by accident, it becomes restricted to class 2. It will keep its CD4 and then downregulate CD8. If it just by accident can bind to class 1, it'll keep its CD8 and then downregulate CD4. So that's instructive. Why it's called instructive is because whatever it binds to, is able to bind to, tells this thymocyte what to do as far as CD4 or CD8. Keep CD4, get rid of CD8, or keep CD8, get rid of CD4. Okay. Now, stochastic means randomly we either downregulate CD4 or CD8. So the thymocyte just randomly downregulates CD4 or CD8. You can see the result of that in the middle. Your CD40 low, CD8 high. That's an 8, not a G. And then on the one, if you just downregulate CD8, your CD4 high and CD8 low. And then go figure out the rest. All right. Everyone figure that out? Everyone figure this one out? I know there's some weird sounds going on out there, but this is not kindergarten or first grade. Let's deal with it. All right, so what happens in this situation? Yes. Go ahead.
Within the pool of cells, some of them will just randomly downregulate CD4.
Yes.
So that
they will only be able to bind to MHC class 1 to become CD8 positives, and then they would end up
So they downregulate CD4, right?
to their A high.
So that means if they bind to class 1, they're rescued because they still have CD8. But if by accident they made a TCR, which is class 2, restricted, it doesn't have the CD4 anymore, so it can't bind very tightly, so it dies by neglect, in essence. So it undergoes apoptosis. That's correct. And why don't you just take the opposite?
And then the opposite thing, CD8 gets downregulated, so the CD4 is combined with class 2 to be rescued, or they would die by neglect in apoptosis.
Right. All right. So that's stochastic. The one which is currently favored, though, is this one, the kinetic one. And I'm not sure why it's called kinetic. At any rate, so what happens is you have CD4. This should be a positive here. There's a mistake in your book. This should be a positive. Should be double positive this stage. So put a positive there. And you can tell they're both there anyway. And then in this situation, CD8 is down regulated, just as a controlled feature. It's down regulated. So now you're CD4 positive and CD8 low. And then again, you have your antigen with class 1 or class 2. And in this situation, we have either a discontinuous or a continuous signaling. Continuous signaling if you're class 2 restricted. Discontinuous if you're class 1 restricted. And it turns out what rescues these cells is IL-7, presumably. And go ahead and make sure you understand this, and then I want someone to explain this to me in your own words, what the kinetic signaling model is, which is currently the favored model for how you decide whether you're CD4 or CD8. Remember, it's a model. It hasn't been definitively proven to be the case. But it's the currently favored one. And immunology, as you know, is dynamic and in a state of flux. Who knows what it really is. Your textbook, by the way, is copyrighted 2019, so you're living in the future, which is kind of cool. If we could really do that, you could tell us what stocks to buy, and then that'd be really nice. All right. Who could just tell me, more or less repeat what I told you, what's going on with the kinetic model? Someone volunteer. You make eye contact with me, I'll assume that that means you raised your hand. Yes, thank you.
So you start with a CD4.
Remember, this is a mistake. This should be double positive at this point.
And then once your TCR is engaged, CD8 is downregulated.
Right.
Produce CD4 positive and CD8 low. So,
Right?
and then after, so either an antigen, the cell is engaged with either an antigen or an MHC class 1 molecule to produce a CD8 positive T cell.
Right, but that only happens if you get disrupted binding, because remember you downloaded the CD8, so you don't get really tight binding, just a weak binding, and you're rescued by IL-7, then you'll become a CD8 positive cell, correct? Go ahead, perfect, go ahead.
And then in the case where I started getting antigen class 2, then the CD4 T cells.
Right, so this way you get uninterrupted, continuous signaling. That sends a signal somehow transcriptionally to downregulate, to keep rather CD4, not to upregulate CD8. So since it's the most complicated model, of course, this being immunology, that's the one which is favored, and it probably is the one to correct. Now I typed something in here, and the reason I typed this in is because this is a treatment, not a completely new treatment, but certainly a treatment which isn't that old, for multiple sclerosis, one of the most common autoimmune diseases. And sphingosine 1-phosphate can appear on lymphocytes, and it helps lymphocytes leave the lymph nodes, leave the bloodstream. And there's a monoclonal antibody which is produced, which binds to sphingosine 1-phosphate. And when it does that, it prevents the lymphocytes from infiltrating the central nervous system, and therefore can abate or downregulate the effects of multiple sclerosis, that is the demyelination in the central nervous system. So that is certainly a treatment. The downside to that is, the side effect of this treatment is that you are susceptible to lots of infections, potentially, especially in the brain. But, at any rate, certainly it has improved the prospects and the lifestyle of people, with some people with multiple sclerosis. All right, now here's a really nice experiment right from your book, in this case, well known. This is an experimental procedure, it's something wrong with the E there, for fetal thymic organ culture. So they took the thymus out of a fetal mouse, they placed it in culture, FTOC, kept the thymus alive, all right, with growth media, etc. And what they then did is they took cells which were transgenic for recognizing the OVA peptide in the context of class 1 H2KB. I've typed that in for you. So you know exactly this is class 1 because we are the K locus. And by the way, is that centromeric or telomeric, this K locus? Centromeric or telomeric? I'm helping you review for next week. Is it centromeric or telomeric? Centromeric. And what haplotype is it? B. All right, we can know that because it's lowercase b. All right, so it's transgenic. You know what that means by now. And then on the C-TEC, so what does C-TEC stand for? Cortical thymic epithelial cell, all right? And then what they did is they didn't put any peptide, and let me tell you the secret of this is, class 1 molecules cannot be expressed in their normal form on the cell surface unless they bound to a peptide. However, they can be expressed in a disarrayed form on the cell surface without the peptide in the binding clefts. This is a disarrayed class 1 molecule. Here's the beta 2 microglobulin, here's the class 1, but it's not orderly in its organization because it's missing that processed peptide. In the second situation, we put an OVA peptide. Remember, this is a class 1, so about 8 to 9 amino acids long, which had low affinity for this TCR. And once you add this pre-digested OVA peptide, it can bind extracellularly to the binding cleft and lead to the correct organization and presentation of our class 1 molecule. Note the difference between the none and the low affinity. So it's an intermediate or low affinity type situation. And the third thing they did in this fetal thymic experiment, was to add high affinity OVA. So this means the TCR can bind with high affinity to this particular 9 amino acid piece of O-valbumin. And now tell me what happened. Okay. figure out what happens. It's a nice experiment to show us positive and negative selection. Death by neglect, positive, negative. All three phenomena are described. Remember, I'm having embroidered for all of you junior immunologists, which you can put on your sweaters, your letter sweaters, or what have you, if they have such things anymore, or on your varsity jacket. And if you don't, if you don't show me on the next exam, the one after that, they deserve your embroidered patch. I won't give it to you. So, go ahead. I'm not actually making embroidered patches for us. I'm not that insane, all right? So, all right, let's try number one here. Here's our CTEC. Class, do this in unison. What does CTEC stand for? Right, and it's our class 1 molecule. Is this a normal class 1 molecule? Why is it abnormal? Because it doesn't have what? The process peptide. Therefore, our transgenic thymocyte, which is OVA specific and class 1 restricted, okay, it's nothing for it to bind to tightly because it doesn't have the process peptide. Therefore, this thymocyte will die from what? Neglect, because it fails positive selection. There's nothing for it to recognize. Okay. What about the middle one? Again, this is our Goldilocks here. This is what we're just showing here. So what's the middle one? The OVA binds. Doesn't bind greatly, but it binds well enough. So we now know the TCR can recognize the class 1 molecule that is its own, but not so much the self-peptide that'll be self-reactive. Remember what molecule it upregulates if it binds just right like this? Begins with a B. BCL2, which prevents what? Apoptosis by inhibiting what string of enzymes I told you? Caspases. We'll have more to say about that. And does this undergo positive selection and survive? Yes. What about the third situation? Now we have the OVA this TCR really wants to bind to. That's the right specificity. It binds real tightly. And that means this thymocyte's terrified that in the periphery it's going to encounter all this OVA, right? And therefore that sends a strong signal now to undergo apoptosis because this is a self molecule recognized as such, a strong interaction, and therefore we get negative selection taking place and the thymocyte kicks the bucket. Now, this is a slide from your textbook I didn't originally include in your slide set, but it's in your textbook, figure 8.7. I crossed out the numbers here because they're quite confusing and misleading, so I just crossed them out, put a white rectangular box to hide them because they're confusing, so don't worry about those. But I want you to examine this for a little bit. This is really a nice summary of what current research, and just what we know current research, might not be what happens in a year or so, but from our current understanding of what's going on, the different types of T cells, mature T cells, which will emerge from the thymus under 4 different types of binding circumstances. So I want you to read that right now. Again, this is right from your textbook, figure 8.7. Again, I told you to ignore the numbers on here, they're misleading. There's something you haven't seen before. All right? I want you to point out specifically what you haven't seen before. That's really why I showed the slide. All right, let's go through this again as a learning experience. Collectively, communally, become communitarians. Low. Fail to be positive selected. Our favorite expression in this case is you don't recognize anything, you die by what? Neglect. Again, a great title for a horror movie. Death by neglect. All right. Intermediate. We just looked at that. You get somewhat of an interaction, not too strong, so you would proliferate in the periphery against this molecule, but enough to induce BCL2 so you don't undergo apoptosis, and this is positive selection. However, what if you have higher, slightly higher than intermediate? People now believe that gives rise to a very important homeostatic cell called a T regulatory cell. The T regulatory cell is the other arm of the immune system which down-regulates the immune system. So far in this course, I've emphasized how we turn the immune system on. Obviously, evolution had to figure out some way of turning it off. And one of the ways that's brought about are by regulatory T cells. Like everything in immunology, regulatory T cells were discovered, and then lo and behold, it turns out there's more than one type of regulatory T cell. In fact, there's more than two, but we're only going to talk about two. If you take the seminar, talk about more than two. But there are two main types of regulatory T cells. One's called a thymic regulatory T cell. It develops in the thymus. And there's another type of regulatory T cell which develops in the periphery. That's called a peripheral regulatory T cell. We're not worried about the peripheral regulatory T cells yet. You will shortly. But for the moment, we're just worried about the thymic regulatory T cells. And if you have higher than intermediate but lower than very high, whatever you want to call it, this could be low, this could be intermediate. I would actually use higher than lower, but whatever terms you want to use. Right? So somewhat high, you get regulatory T cells. And if it's too high, you get negative selection and deletion. By the way, thymic regulatory T cells used to be called natural regulatory T cells, and you'll still see that term, but it's no longer in vogue. And the ones which developed in the periphery used to be called induced regulatory T cells. That's also no longer in vogue, and now called peripheral regulatory T cells. That's just making immunology more complicated. They keep on changing the terminology as well, for reasons I'm never quite sure. Well, I guess natural and central don't mean that much. All right. So now, peripheral induction of tolerance in T cells. Is it possible, even though you have AIRE, there might be some antigens, self-antigens, which are not expressed in the thymus? What would your innate thought process tell you? Would there be potentially some molecules in your genome which just AIRE didn't allow to be transcribed, and therefore were not expressed in the medulla or in the cortex, and therefore negative selection against these self-peptides did not take place in the thymus? Isn't that possible? I'm directing you to the answer which is only possible. Do you agree with me? Yes. So what's a system to do if there's a molecule which can be expressed in your body but wasn't expressed in your thymus? How do you become tolerized to that so you don't react against the self molecule? Because if you fail to do that, you'll have an autoimmune state. And sometimes that happens, unfortunately. We all know people with autoimmunity. I guess I would imagine most of you do. But most of us do not have autoimmunity. So we must have evolved a way to become tolerized to these self-antigens outside of the thymus. And indeed there is. Experimentally, the following situation can arise. Here's a mature cell, T cell, say helper cell. Okay. And we're in the periphery now. Here's a dendritic cell. Dendritic cell, let's say class 2. All right, and here's the TCR. All right, so now the TCR engages this dendritic cell and can see is class 2 restricted, and whatever antigen you want to make up in your head specific. Whatever antigen you're thinking up in your head specific. Okay, let's make something up. Let's say glucagon. Just to have the opposite of insulin, let's say glucagon specific. Glucagon, you know, raises blood sugar as opposed to insulin lowering blood sugar. So let's just do the opposite. So glucagon, specific, class 2, restricted. All right? Now that should activate this particular mature T cell in the periphery. But that's only the first signal. Turns out you need a second signal. The second signal is, at a minimum, CD28 has to bind to CD80 or 86. And unless both these things are interacting with their ligands, you will not get activation of this T cell. Now, it turns out dendritic cells and macrophages tend to be cheap about expressing CD80 or 86, unless they, by their TLRs, have recognized something which is dangerous to you and upregulate CD80, 86. So if it's a self molecule, such as glucagon, and a dendritic cell takes the glucagon into itself, it won't be a signal to upregulate CD80, 86. So our T cell, through the TCR, engages the CD28 on the T cell, doesn't find much CD80 or 86, doesn't get that second signal, and in that case, you get a signal to this T cell to energize, the opposite of energy. Energize, the opposite of energy. Energize becomes energic. That means it becomes incapable of becoming activated. Lassitude. Energize, great Scrabble word. Only the immunologist would get that. So you become energized, you will not be able to become activated. The problem with this hypothesis is, and it's a pretty good one, is that many dendritic cells seem to have CD8086 expressed. So maybe that explains why autoimmunity evolved, because for some reason these sneak through and they encounter a self-antigen or a dendritic cell with enough CD8086. You get this T cell now to get both signals, and then it starts to become active against self. That's one possible explanation. I'm not saying it's a universal explanation for how autoimmunity might arise. So I'm not saying this is an absolutely perfect means by which we can exclude self-reactive T cells. And we know that's the case because for sure we have autoimmunity. Now it turns out these are two signals you need. This is the second signal. There's a third signal you need, though, for the T cell to be activated in the periphery. Third signal is that you need to have some cytokines produced by the dendritic cell, cytokines such as interleukin 12 and others, which are needed for the T helper cell to become fully activated. And we'll talk about these cytokines. It's on the syllabus. When I have the topic cytokines, we're not going to deal with that really today. So, as you can see, it becomes complicated. All right. And this is just exactly what I said, the three signals that you need to get this cell, T cell going. And if you don't have that co-stimulatory interaction, which is CD28, just like I drew it here, CD8086, then you'll get anergy, as I've typed in here. All right. Now, regulatory T cells. Well, they can be produced in the thymus, as I explained briefly in one of our slides, and also can be produced in the periphery. How they're produced in the periphery, we will learn. Just take my word for it. They can be certainly produced in the periphery, certainly experimentally. Now, how do regulatory T cells operate? Well, let's draw that on the board. By the way, not to give you an off switch in your brain, but from here on in, what I'm going to say is on the third exam, not on the second exam. So this is not for you to flip a switch in your head and break the circuit. All right. We still have almost over 15 minutes to go. All right. What are regulatory T cells? How do they do? Okay, what am I drawing here, class? DC, a dendritic cell. What's it have right in the middle? Just put a nucleus in it, like a donut, an ill-formed donut. And true or false, this information is going to be asked of you on the second exam. So don't email me and ask me that. It is not on the second exam. However, is it on the final exam? Yes. So first of all, pay attention. All right. So I have class 1, I have class 2 on the surface here. I have 80, 86, and other things as well on the surface of my dendritic cell. Let's do a class 2 restricted T cell. I like them better. So here's the T helper cell. So class 2 restricted. Okay, here's CD28. This is the T helper cell, so it's going to recognize all this mess on the dendritic cell and start to become active. Now, I'll make this a class 2. Does a dendritic cell express only one class 2 molecule on its surface? Does it express zillions of copies of class 2 molecules on its surface? Zillions of copies of all the class 2 molecules in that person's repertoire. So therefore, another cell can bind to the same spot. This time, it's a regulatory T cell. And a regulatory T cell binds to the same dendritic cell. And through its TCR. But instead of having CD28, regulatory T cells have something called CTLA4. CTLA4 binds to CD8086. We have CTLA4 on a regulatory T cell. CTLA4 binds to CD8086, and by the way, CTLA4 is what was given the Nobel Prize this year for to the person who discovered it. It's Professor Allison from Texas. He discovered CTLA4, and so just keep that in mind. So now we have juxtaposed a T helper cell and a regulatory T cell, and they're near each other. So now they can have influence on each other because they're juxtaposed. They are now near each other enough so they can actually have a physical contact or close enough of a physical contact to have an effect on each other. And if the regulatory T cell produces anything, it's so close to our helper T cell, that can affect the T helper cell in a way that's called a paracrine effect. Paracrine means short distance effect. It works over a short distance. Paracrine. All right, so how do regulatory T cells downregulate the immune response? In many ways. It's like Elizabeth Barrett Browning. I laugh at my own jokes. All right, anyway, regulatory T cells via CTLA4 block CD8086 on dendritic cells. And it turns out CTLA4 has a higher affinity for CD8086 than CD28. Now I presented you a problem here. I just said that CTLA4 binds with higher affinity on CD8086 than CD28. How in the world would you ever get then activation of this helper T cell? Seems to me that you'd always put the brakes on the system, you'd never go anywhere. Allegra?
Can the T regulatory cell upregulate the T helper cell?
So say that again?
Can it upregulate instead of downregulate?
Can what upregulate?
The T helper, like send some cytokines that are now awake.
So it can upregulate what?
The T helper?
Yeah, but if the regulatory T cell here via CTLA4 is binding to high affinity to CD8086, it may muscle out the ability of the CD28 to bind to 8086.
Yeah, but I was thinking that maybe cytokines can counteract that.
Okay, that's one idea. One idea. Yeah?
Are T regulatory cells expressed in much lower levels than T helper cells?
Yes, and so they may come into the scene later, if you will. So they're late to the scene. and therefore this interaction may not take place temporally at the same time, and therefore the helper phenotype will emerge before the brake is put on the system. You won't get any more activation because now the CTLA4 dominates. So yes, well, we're not really sure, to be honest with you, but that's one possibility. And by the way, not too surprisingly, one of the cancer chemotherapies, again, is ipilimumab. Ipilimumab is a monoclonal antibody which binds to CTLA4, preventing it from putting a brake on the system. So that's one way that helps fight cancer, because you don't put a brake on the T cells. Okay, 2. Read this one. There's an enzyme produced by dendritic cells called IDO, indoleamine 2,3-dioxygenase, which breaks down tryptophan. So what happens is a T helper cell binds, regulatory T cell binds that, stimulates the dendritic cells to release IDO. IDO then bathes the T helper cell with this enzyme which destroys tryptophan. Tryptophan is highly utilized in helper T cells and cytotoxic T cells for that matter, and therefore it cuts down on T cell proliferation. So this is definitely another pharmacologic intervention people thinking about. Regulatory T cells can do this in a boring way. They can produce 2 cytokines called TGF. TGF stands for, thank God, it's almost Friday, transforming growth factor beta. Right, so TGF, transforming growth factor beta, a really important cytokine involved in healing and it's anti-inflammatory. TGF beta is anti-inflammatory. It's involved in the healing process, as is IL-10. Introduce you to 2 cytokines today, TGF beta and IL-10. Okay? So it's another possibility. And again, if this thing's pouring out IL-10 and TGF beta, it's going to be by paracrine effect, be able to influence this helper T cell and downregulate its activity. I didn't mean to do 2 at once there. All right, so another thing it can do, regulatory T cells can produce 2 molecules called perforin. Perforin is a protein which polymerizes on its target cell. What do you think perforin does? It's a protein which polymerizes on a target cell. Perforin, what do you think it does to that target cell? It perforates it. So it cuts a hole in it. That's sort of like the MAC attack. Well, remember what the MAC attack is? Membrane. Right, okay, so this is perforin. It's another form of that, if you will. It drills a hole in our poor helper T cell. That doesn't kill it. What kills it is the regulatory T cell also releases molecules called granzymes, which digest, sort of like alien. So it has perforin, which drills a hole in the helper T cell. It then injects granzymes into the helper T cell, and just like alien, it consumes the host by breaking down its integrity, cellular integrity. I don't know if you guys saw Alien, but creepy to the nth degree. All right, and turns out finally, competition. There's always competition in life. Regulatory T cells express CTLA4. Okay, I tell you that combined to CD86. In addition to that, they also express a receptor for interleukin-2. And interleukin-2 must bind to a receptor, which has 3 components: the alpha receptor, the beta receptor, and the gamma receptor. And unless you have all 3 components of the interleukin-2 receptor, you don't bind interleukin-2 very efficiently. Helper T cells only produce the right type of interleukin-2 receptor after they start to become activated. And therefore, helper T cells are not capable of responding to the IL-2 until after a while. until after they upregulate the IL-2 receptor. And one of the components of the interleukin-2 receptor is called CD25. However, regulatory T cells always have a high affinity interleukin-2 receptor. So here's what happens. T helper engages the dendritic cell. T helper produces interleukin-2. Interleukin-2 is normally an autocrine. That means it stimulates the cell that produces it. So that's even more limited than a paracrine. But the regulatory T cell outfoxes the helper T cell because it has a receptor for IL-2 right from the beginning, which is high affinity for IL-2. So the helper T cell starts producing IL-2. It doesn't yet express the high affinity form of IL-2. The regulatory T cell already is equipped with it, so it steals the IL-2 from the helper T cell, steals it, because it has the high affinity version, expresses CD25. In fact, one of the ways we identify regulatory T cells is that they have high expression of CD25. It's one of the ways we can, by flow cytometry, isolate regulatory T cells. One of the ways by looking for CD25. And the regulatory T cells will snag the interleukin-2, steal it from the helper T cell, and prevent the helper T cell from becoming active. So those are the various ways that regulatory T cells carry out their function. All right, so T cell activation, I already told you you need 3 different stimuli to get the T cell going. And now look at this slide. Aren't you glad this isn't on the second exam? This is the family of T helper cells. I want you to look at them right now. Some of them we've talked about, some of them we have not. Let's look at them. By the time you leave this class, you'll know every one of them. And so we're going to start with a naive CD4 cell, and we're going to somehow get polarizing cytokines, so they end up having what's called a master transcriptional regulator, being upregulated. They're transcription factors. And that will determine the fate of this early T cell, either to become a red cell, T helper 17, T helper 2, T helper 9, follicular helper T cell, T helper 1, or T helper 22. And yes, every day we're probably going to get more different additional subtypes of T helper cells. Honest with you, the only ones we're really going to talk about are peripheral regs. I'm not going to talk about thymic regs. We're going to talk about T helper 17 in this class. We're going to talk about T helper 2 in this class. We're going to skip T helper 9. We're going to talk about follicular helper T cells for sure. We're definitely going to talk about T helper 1. And we're not going to talk about T helper 22. So on this cast of characters, we're not going to talk about T helper 22. We're not going to talk about T helper 9, but every one other one we are. And you can see, as you go, this is a nice summary slide. You can tell the type of cytokines each of these different phenotypes produce. Some of them are obvious. T17 cells produce IL-17. T9 cells produce IL-9. T22 cells produce IL-22. Okay, and this tells you the effector function. And finally, the last column of all, our row, tells you if there's too much activity by this type of T cell, what disease is. Hey, I can see you talking back there, right, is involved. So the cells are too active. When I was in 9th grade, I talked my way into a C in class because I gabbed the whole way through. The teacher was so annoyed with me that he gave me a C. Only C I ever got in junior high school. All right, so to this day I'm embarrassed about that. All right, at any rate, this is what happens with rolling disease. Plus I passed notes. That's before you could text. So that was even worse. And he actually took one of my notes away from me, so made me more embarrassed. All right, and read them to the class, which I'm still turning red thinking about, like, just as a warning. All right. So these are the cast of characters as far as the different types of T helper cells. And by the way, I see everything I was doing out there, so there is no escape. Who falls asleep, who's not paying attention, who's laughing, who's talking, and who's planning for the weekend. All right, so we're going to learn all these, again, cells with the exception of T9 and T22. Not that they're not important, it's just not enough time. And on next Tuesday, we're going to talk about generation of memory T cells. Before you go, I want to show you how complex memory T cells are. This is a simple rendition of different types of memory T cells. This is a more complex rendition of memory T cells. And this is a more complex explanation of memory T cells. For the third exam, you will have to know this. All right, so have a great weekend. Remember, we have a review session on Wednesday night, 6 to 7:30. Okay. It's on canvas.
Did you say so this lecture as a whole will be?
No, I did not say that. I said this lecture will be, except where I started talking about I need to make sure. I was very careful when I said that.
Okay.
Let me make sure, and now you got me mixed up.
Oh, let you make sure. Okay, I didn't hear you.
Said your responsibility for the second exam. I know this.